Very good morning all. So let us move to the one more concept in the nanotechnology, that is the phytonanotechnology. Okay. So before that we should know regarding the nanoparticles. Already I told regarding the nanotechnology and nanoparticles. So nanoparticles are the particles which lie in dimension between 1 to 100 nm. So their dimension is between the 1 or up to 100 nm. Nano derived from the Greek word nanos, which means dwarf or extremely small. So very much small particles we call as the nanoparticle that we are going to measure in a nm. It can be used as a prefix for any unit to mean a billionth of that unit. Means if you take one part of the billionth, that is the nanometer. For example, nanosecond, that is nothing but billionth of a second. In 1 second you make 1 billion, so that one part of the billion is the nano, nm. For example, one more, nanometer, billionth of a meter. In 1 meter you make the 1 billion part, so that one part is the 1 nanometer. Again same as for the nanoliter. Like this is very small particles that we going to measure in a nanometer. They consist of micromolecular materials in which the active ingredients, drug or biological active matter is dissolved, entrapped, encapsulated, absorbed or attached. So the nanoparticles which contains the very small material, so that may be contains the ingredients. So the ingredient may be dissolved or entrapped or encapsulated. So the nanoparticles, because of the micronutrient, it will be dissolved or it may not be dissolved. So this is the methods for synthesis of nanoparticles. If you want to synthesize the nanoparticles, we have to use the various methods. It may be chemical method or physical method or biological method. By using 3 methods, you can produce the nanoparticles, and with the help of nanoparticles, you can produce the different products and the byproducts. Those are useful for the human beings. That I told, some plates, glasses or any other materials, suits or we call as the fabrics. All these things are produced by using the nanoparticles, which act as anti-adhesive, antioxidants. Okay, anti means antibacterial, antiviral, like all these properties played by the nanoparticles. So by using these techniques you can produce the nanoparticles. Physical method means how you going to produce by the physical method. It will be require time and energy consuming synthesis at high temperature and pressure. If you using the physical method, you have to use the high temperature and pressure. Chemical method means it's simple, inexpensive and low temperature. Use of toxic reducing and stabilizing agents make it harmful. So if you use the toxic reducing material, so it will make as a harmful. Green method, it is very easy, efficient and eco-friendly. Okay, so the plants and the other microorganisms comes under the green method or biological method, which will be eliminates the use of toxic chemicals, consumes less energy and produce safer products and byproducts. By using green method, you can produce certain nanoparticles which are safer, produce the products and byproducts. Like the green synthesis, it may be by the microorganism or plants or any crops, that we call as the green synthesis. Then phytonanotechnology. So the phyto refers to the plants. Okay. So the nanoparticles are going to be produced by the plants. Those are very much useful for the human beings. That is nothing but the phytonanotechnology. It is actually the synthesis of nanoparticles using fresh plants or plant extract. You can use the fresh plants or any parts of the plants or any extract of the plant. By using that, you can produce the nanoparticles. Plant derived nanoparticles produced by readily available plant materials. Okay, it may be any plant material. The non-toxic nature of plants are suitable for fulfilling the high demand. So you have to use the non-toxic plants. for production of the nanoparticles with applications in the biomedical and environmental areas. Recently, successfully synthesized gold and silver nanoparticles using the leaf and root extract from the medicinal herbal plant, that is Panax ginseng. So gold nanoparticles were synthesized and again silver nanoparticles were synthesized by using the leaf and root extract of the medicinal plant Panax ginseng. So comes under the phytonanotechnology. Additionally, various plant parts that may be stem, root, their extract used for the synthesis of metal nanoparticles. Okay. It has been proposed that proteins, amino acids, all these compounds and polysaccharides have significant roles in metal salt reduction, and furthermore, so for the chemistry, that is for the reduction of the metal things, they used the vitamins, amino acids, etc. Act as capping and stabilizing agents for synthesized nanoparticles. All these vitamins, proteins plays an important role for the synthesis of nanoparticles, and these will be act as a stabilizing material. For instance, the hydroxyl functional group from polyphenols and the carbonyl group from proteins of Carolina officinalis extract could assist in forming and stabilizing gold nanoparticles. So the polyphenols of the Carolina officinalis and other plants extract that will be helpful forming the means act as the stabilizing agents for the gold nanoparticles. Reports also suggest that different mechanisms for synthesizing nanoparticles exist in different plant species. Okay, so some reports what they suggested means so many different mechanisms are there for synthesizing the nanomaterials that will be present in all different plant species. Notably, dicot plants contain many secondary metabolites. Secondary metabolites act as a stabilizing materials for synthesis of the nanoparticles. That's why all the plants have the metabolites and certain stabilizing compounds. Dicot plants which will be produce the metabolites, secondary metabolites, which helpful for the production of the nanoparticles. So these are the some stabilizing agents or stabilizing particles present in a different plant species. For example, eugenol, the main terpenoid of Cinnamomum xylonisum, was found to have a principal role in the synthesis of gold and silver nanoparticles. So these plants plays an important role for the production of the silver and gold nanoparticles. So these are the plants microorganisms which produce the nanoparticles: leaves, fruit, stem, peel. Okay, metal nanoparticles produced by the microorganisms, yeast, fungi, bacteria, all that we call as the microorganisms nanoparticles were produced. Phytochemical screening. It refers to the extraction, screening and identification of the medicinally active substance found in plants. So some active substance present in the medicinal plants, so that extraction, screening will and identification will be done by this method. Some of the bioactive substance that can be derived from plant, that is the alkaloids, carotenoids, tannin, etc. Color change or precipitation formation is the indication of presence of these phytochemicals. So phytochemicals are present in the plants. How it will be identified, how it will be screened means due to the color change or precipitation occurs, then we come to know the phytochemicals are present in the plants. Preparation of plant extract for phytochemical screening. You have to first prepare the plant extract. How you going to prepare the plant extract means take fresh plant. An ethanol extract of sample can be prepared by milling 50 gram of the desired part plant in an electric blinder. Okay, then mixed paste of plant with 200 ml ethanol in conical flask by shaking it with hand and placed for 3 days or by mixing it with magnetic stirrer. Now filter the ethanoic extract of plant. Like by using this method or procedure, you can extract the plant, then you come to know the phytochemicals are present in those plants. Place the filter on rotatory evaporator at 60 degree centigrade to remove ethanol. The extract will be concentrated by placing it on water bath at 75 degrees centigrade. Now the sample is prepared for the phytochemical screening. Okay, this up to this you have to follow the procedure, then you can extract the plant extract. Preparation of plant extract for synthesis of nanoparticles. Again one more, that is the you have to prepare the plant extract for the synthesis of nanoparticles. To prepare the plant extract, take desired part of plant, it may be leaves, wash it properly with tap water. Now sterilize it by washing 2 to 3 times with deionized water. Dry it now room temperature. Boil 40 gram of leaves with 200 ml of deionized or distilled water for 20 minutes at 70 to 80 degrees centigrade. This is the method for the extraction of plant for the synthesis of nanoparticles. During boiling, color of solution changes to green yellowish. Okay. Cool the resulting extract at room temperature. After that, the yellow colored extract must be filtered with Whatman filter paper. Now, cover the filtrate with aluminum foil and store it in refrigerator for further use. These are parts you can use for the plant extract: root, leaf, coir, gum, seed, fruit, etc. Use of plant extract in nanoparticle synthesis. In producing nanoparticles using the plant extract, the extract is simply mixed with the solution of the metal salt at room temperature. Okay, you have to mix the plant extract with the metal salt at room temperature. The reaction is complete within minutes. Nanoparticles of silver, gold and many other metals have been produced this way. Most of the silver, gold or any other metal ions going to be produced by this method. The nature of the plant extract, its concentration, the concentration of the metal salt, pH, temperature, contact time are known to affect the rate of production of nanoparticles. So the proper condition you have to give for the production of the nanoparticles. Their quantity and other characteristic all you should know before production of the nanoparticles. So this is the plant which we use, leaf plant extract solution, metal salt. Where when you produce the nanoparticles, the color will be changed, which shows the different color, that we call color change shows nanoparticle formation. Characterization of nanoparticles. What is the characteristic? Nanoparticles are generally characterized by the size. Of course, it is size, shape, surface area and dispersity. All these characters present in nanoparticles or properties. A homogeneity of these properties important in many applications. So all these properties or character is present in the nanoparticle, then it plays important role in the different fields. The common technique of characterizing nanoparticles are as follows. UV visible spectrophotometry means all these you have to use for the study or the characterization of the nanoparticles. Dispersive spectroscopy, all these machines you have to use, all these microscopes you have to use for the identification of the nanoparticles. What are the applications? So silver nanoparticles means we call as antimicrobial, anti-protozoan, anti-cancer, antifungal. Okay, for those disorders or a diagnosis of these diseases, we have to use the silver nanoparticles because it acts as antimicrobial, anti-protozoa and all these things. Gold means antimicrobial, palladium antibacterial, copper antibacterial, anti-cancer. Then selenium, that we call as anti-cancer. All these metal nanoparticles have the application in various fields. What is the advantage of phytonanotechnology? So the phytonanotechnology has provided new avenues for the synthesis of nanoparticles and is an eco-friendly, simple, rapid, stable and cost-effective method. Okay. Phytonanotechnology has advantages including biocompatibility, scalability and the medical applicability of synthesizing nanoparticles using the universal solvent water as a reducing medium. If you produce the silver, gold nanoparticles or any other metal nanoparticles, those have the medical application because the property which are present in the gold and silver nanoparticles, those have the low bioavailability, low temperature, low cost, all these things. Okay, so up to this regarding the phytonanotechnology. So this question will be asked for 5 marks regarding the phytonanotechnology. So all these things you have to write in that phytonanotechnology. Okay, thank you.